Hello, my name is Kylie Weis. This summer, my project has been it's not working. Okay. This summer, my project has been to be or not to be, using pollinators to induce soybean hybrid vigor. So my mentor has been Nicole Saluga and Dr. Margaret Frank's lab. Soybeans, or scientific name glycine max, is a commodity crop in the US with 88 million acres planted this year and $75 billion worth produced in 2021. Soybeans are often used to produce vegetable oil, livestock feed, tofu, and soy milk, among many other products. This is why soybean breeding is so important. However, soybean lines grown in the US are often inbred or pure line cultivars. This means that the plants are allowed to breed with themselves and no new genetic material is introduced. This can often lead to yield depression over time and can also open up the opportunity for disease epidemics. However, hybrid soybeans experience hybrid vigor. They often have much greater yields, with reported 16% greater yields than their parent lines. But creating these new hybrid cultivars can often be extremely difficult. It requires hand crosses, which can be very time-consuming and often has low success rates. These low success rates are often due to soybean predominantly self-pollinating. Less than 1% of seeds are the result of cross-pollination in field studies. This is often due to the structure of the flowers. As seen here, there's two sets of petals that cover the stigma and anthers. The stigma and anthers are also located very close together, which allows for easy self-fertilization. These flowers are often difficult for pollinators to find due to their very small size of only 5 to 10 millimeters across. The flowers are often hidden by the large leaves of the soybean plants, and many cultivars have white flowers, which is often difficult for pollinators to see. However, there have been documentation of pollinators in soybean fields. Bombus impatiens, the eastern bumblebee, and Apis mellifera, the western honeybee, are the two commonly found pollinators in soybean fields. For this study I did this summer, we used Bombus impatiens, the eastern bumblebee. Bee vision is very important to them being able to see the flowers to be attracted to them. Both Bombus impatiens and Apis mellifera have 3 photoreceptors, one for blue, green, and UV. Multiple bee species are documented having a preference for purple-colored flowers, and this is likely due to that color falling within their vision range. While many cultivated soybeans have white flowers, Glycine soja, a wild relative of soybean, has purple flowers. This image on the right shows the difference between Glycine max and Glycine soja. Glycine max often has a growth phenotype that is much better suited to field growing conditions, while Glycine soja has a vining growing type and less nutritious seed pods than soybeans. But luckily, they also have purple flowers. The purple flowers are caused by the W1 allele, which encodes for flavonoid 3'5' hydrolase. This hydrolase is part of the anthocyanin and delphinidin pathway. This is a molecule delphinidin. It is an aromatic pigment that is pH dependent and produces that purple color at low pH. This W1 allele in many cultivated soybeans has a 65 base pair duplicated insertion that truncates the protein and does not allow the completion of the pathway in the production of the purple pigment. My objective has been to assess pollinator attraction to soybean flowers of various colored phenotypes to assist in developing soybean lines for a cross-pollination hybrid breeding system. My hypotheses are 1, bumblebees will more likely to visit purple soybean flowers when given a choice between purple and white. 2, of the purple flowers, the flowers that are darker purple will receive more visits than the lighter colored flowers. My methods have been, we have tested 9 transgenic soybean lines of varying degrees of purple color. We conducted cage choice trials of Bombus impatiens with one purple flowered plant and one white flowered Williams 82 plant. We phenotyped the flowers using image analysis software, and we calculated average number of visits per flower for both white and purple, created a white to purple ratio, and compared it to phenotypic data. The transgenic soybean lines were created with a construct with the functional W1 allele from Glycine soja and the native W1 promoter. The University of Wisconsin's Crop Innovation Center performed these transformations. An agrobacterium was used to randomly insert the W1 allele into the white cultivar Williams 82. The flowers were photographed using a dissecting scope, and they are all photographed in the same light conditions. We then phenotyped the flowers using ImageJ software. We traced the flowers, that line of flowers, to only include the petals. We then used the color histogram tool to find the RGB values. These RGB values were used to find the red to blue ratio and the average RGB value, which is an indicator of perceived brightness of the flowers. The following phenotypes are as follows. We had a range of flowers from dark purple to medium purple to light purple in Williams 82. To understand a little bit more about the RGB averages, 0 is indicative of black and 255 represents white. We set up the trials using a mosquito net tent. We'd have one hive of Bombus impatiens, and we'd have 2 plants, one purple and white in each tent. We'd also have 2 counters, one for each plant. We'd open up the hive. We'd record which bee, we'd record the color of the flower that the first bee visited. Then we'd also count the number of visits per plant for 5 minutes and then an additional 5 minutes. These were repeated 3 times for each of the 9 plants. We noticed some interesting bee behaviors while doing these cage trials. Bees would often go to one flower and then stay on that plant, visiting all flowers on that plant before moving on to the other plants. We also noticed they would open closed buds and tear petals off the flowers. And then here is a short clip of the behavior on the plants. As you can see, there is many bees on the same plants. This could often make it difficult for us to count and keep track of the exact number of visits. Our results are as follows, with this chart showing the visits per flower in 5 minutes. Initially looking at this, it doesn't seem to be a clear winner of number of visits to white or number of visits to purple. This was confirmed with a T distribution with a high p-value. They do seem to visit white and purple equally. We confirm this with a correlation matrix. In this correlation matrix, 1 equals perfect correlation, 0 equals no correlation, and -1 equals perfect negative correlation. We did not see any significant correlation between the visitation ratios and the phenotypic data or color of the flowers. However, we do see some strong correlation here between the red to blue ratio and the RGB average. This indicates that the bluer flowers are often the darker flowers. However, when we look at a different metric, when we no longer look at the number of visits in a certain time period, when we only look at that initial flower color that the bees first visited, we do start to see a pattern. We see moderate negative correlation between the red and blue ratio, which means that the bluer the flowers, the more likely they are to visit purple first. We also see moderate negative correlation between the RGB average. This means that the darker the flowers, the more likely they were to visit purple first. Interestingly, we see almost no correlation between the first visit color and the amount of visits within 5 to 10 minutes. When we divide the flowers into the darker blue flowers and the lighter, more pink-colored flowers, we see an even farther divide, where the darker flowers are preferred over the white flowers. However, the lighter purple flowers almost seem to have a 50-50 split between white visitation. This brings up the question, what truly is the best metric for bee color preference? Is it the number of visits in a certain time period, or is that initial flower that they go to really indicative of their preference? And I like to use the analogy, if you were given 2 desserts, you would likely pick the one that you find most appealing first. But if you're given time to try both, you will likely try both. Future directions. For the study, it'd be interesting to look at color preference from a distance, as bees would be flying over a field and be able to see the color. Test if purple flowers are more attractive than white flowers in field conditions, or there are other native wildflowers in competition with these flowers. Test preference of other pollinator species, such as Apis mellifera, and find the W1 allele copy number, genome location, and check for homozygosity to see if this is why we saw the different phenotypes. Finally, I would like to thank the institutions that funded this project. I'd also like to thank my mentor, Nicole Salupa, Dr. Margaret Frank and her lab members, Dr. Scott McGart, Paige Muniz, and the McGart Lab, Scott Anthony, the Guterman Greenhouse staff, Megan Truesdale and Olivia Olstein, Blake Myers, Patricia Baldrich, and Ryan Del Precio at the Donald Danforth Center, and Kobe Phillips for helping us with the bees. If you have any questions or comments, please feel free to email me. Thank you.